We are live.
Speak and pause.
We're good on the resolution, by the way.
Speak and pause. Speak and pause.
Why are you saying speak and pause?
Because I'm testing my audio over here. But it's like, it's just very faint. I'm just waiting for Kristen.
May Young sends a heart on the chat of the stream. I'll keep an eye on this for when you do questions at the end. I'll let you know if anyone from the stream has questions.
Well, sounds good.
Oh, cool. The resolution is lit.
Yeah. Yeah, the key thing right now is if Kristen is gonna be able to see this part,
Yeah.
I think she'll be able to hear me. Now I'm like, ooh.
Let me make sure that I guess I should make sure the sound quality is okay. Let me make sure that I guess I should make sure this. Yeah.
I think she'll be able to hear me.
We're good on volume.
Yeah.
Okay, I'm gonna run to the bathroom real quick.
Did it just save your audio recording?
Oh, yay. Okay. All right. I see Kristen. Yay. Hi. Can you hear me okay? Yeah. So I have this. Well, I'm going to screen share with you my slides for this. So I'll do that, I guess, after Barak's introduction. And I'll just check and make sure that you're here. But I guess it'll be on the big screen. Okay, well, I'm not sure if he's going to unplug this or not. Yeah, so he's not in the room yet, but I wanted to make sure that you could hear me. So I'm probably going to be standing here and talking. Can you still hear me? Okay. Yeah, the YouTube stream will have a 30-second delay, but the audio, there's a big microphone on that. And we'll use this for the meeting in a much smaller room. Well, it's important important that you're like here or here. Okay, so I will just. Okay. All right. And then if there's an issue with this at all, just put it in the chat. Because I have my volume, I think, all the way up, but it's not super loud. Yeah, exactly. So it should be loud enough, I think, in the room, and I'll hopefully be able to figure out how to do that. Okay. Alright, well, yeah, Barak's here. Are you gonna need to disconnect my computer?
For what?
Are you plugging in your computer for anything?
No, I have a stick.
Okay. Alright, so you'll get to also, I guess, stream Barak's thing. I guess I'll just leave this like this.
Oh, you got a serious.
I know. It's what the Gordon Lab has.
It's what?
It's the Gordon Lab setup.
Okay, yeah, they have to stream.
Yeah.
So
We're sitting here.
I guess so. I'm surprised anything else on mistakes.
What'd you say?
I'm surprised like there was software you can on.
I found something open source that YouTube said worked. I know. Gordon Lab setup. Their tripod's really nice, but the one problem with it being as flexible as it is that it's really hard to adjust to make sure it's level.
But it looks like that thing from that movie with what's the guy like he's a superhero, but he's a villain. And it's
yeah,
kind
should I do that?
and he's
That's
kind
what
of
I'm
bad.
thinking.
And he has like little girls.
Despicable Me.
Yeah, it looks like the. Oh no. Actually, it might be
You
incredible.
want to share with
Yeah.
Kristen?
Yeah.
Yeah.
So what do they do in Incredibles? It's like one of the villains. Is it Paul?
Yeah.
Oh, yes. Yeah.
So the idea what I'm talking about?
I know what you're talking about. I just can't put a name to it.
Well, the legs look like that. That's what.
I know.
Yeah, it reminds me of that.
Oh yeah.
Yeah.
That was a recent one. Was he in?
There's been one newer than Andrew Cohen. Wait, yeah, it is.
She's here. She can see the screen.
Yeah, so you can see it there. So that's a little video.
All right. About a 20-second delay. Not bad.
20-second delay. Is that for bleep out any any inappropriate words?
Yeah.
Chromatin.
Yeah. Can you guys fill in the front a little bit more?
Go ask the phone first. Everyone kind of moved to the middle.
This is so weird. They don't have any aisles.
Yeah.
It's like last year they ruined the Erlanger those auditoriums also. The ones where you have it Thursdays.
Yeah.
They don't have aisles down the middle.
Yeah, it's just one giant.
Yeah.
It's always like people on
This was built several decades later. They thought they would know.
fix that. Rocks and charge now. Authority beyond belief.
What'd you say?
Like authority beyond belief.
What'd you say?
Everybody's ready to drink her?
Oh.
That's an interesting thing. Tripod is brilliant, so we're not messing with just one single.
Yeah, although it was kind of hard to like make sure it's actually level because it's so flexible.
Yeah. But then once you actually, the easiest just to level if there's some rotating joint at the top.
Yeah, there is.
That one, right?
Yeah, I've got it. I mean, that looks pretty level.
Looks good. This is serious microphone.
I know. I feel like I should be like, making a podcast.
Exactly.
welcome. Thank you for coming. Welcome to Dana King's thesis defense. I suppose if you're not here for Dana King's thesis defense, you are now. Which is not a problem because you're in for a treat. This is going to be great. My job, I guess, to say a few things about Dana before we start. Let me start by showing you this picture. I won't show you this picture.
Maybe go to the down button, I think.
Yeah. Well, this just illustrates the point I wanted to make, which is that I'm inept and Dana is incredibly competent. Thank you.
Now it'll work.
Can I call you back up?
Yeah. No cost to you.
Yeah, so here's Dana in her native environment. All right. So yeah, this shows sort of the two sides of Dana. There's the studious and intense side, and then there's the side where she's always ready for a smile. But there is also another side. Besides the studious side and the smiley side, there's the New Jersey side, which has been an endless source of amusement for me throughout this thing. Dana reminds me all the time. She doesn't say it, but through her words and her actions, she reminds me all the time that she's from New Jersey. And that means that when you're from New Jersey, you're tough. She thinks of herself in this way. So this is her Halloween costume where she dressed. Can you guess what that is?
Tough cookie?
Tough cookie. Yeah, so that's the way Dana thinks of herself. That's the way I think of her. That's the way sort of we all think of her. And when you're tough and you're from New Jersey, that means that you don't back down and you always speak your mind. And that I have one just beautiful illustration of this. And I need to take this. This is from Sam Morris's Twitter feed, for those of you who tweet. She writes back in March of 2002 that she was at the bar, Cold Spring Harbor. That's what the CHL is, the Systems Biology meeting. This is a big meeting where people from all over the world come to discuss gene expression. And Sam is at the bar, and she overhears someone talking about Waddington's landscape, describing it as, quote, ridges and shit, and things going down. Which I think is exactly right. You know me, that sort of sums it up. And Benoit Bruneau says, oh yeah, that's great, I'm going to quote that. Who shall I attribute that to? And it turns out, Dana King, a grad student in Barak's lab. So she's spreading the gospel at Harvard. So that's just an illustration. But when you talk to Dana, you are going to know what's on her mind. It's not going to be hidden. Dana is always going to be front and center in everything that we do. So here she is, right in front and center. No matter what it is, she's going to be front and center. Excitement about science. It's really great to see this kind of enthusiasm. And there she is. So I just want to tell, I guess, a couple stories. We started off in an interesting way. Dana rotated in my lab, and she did a great rotation. She was one of the few rotation students who had really sort of knocked it out of the park as a rotation student. So I kind of knew early that this would be someone great to have in the lab. And then as we talked, she was saying that she really wanted to kind of transition out from what she was doing and become more computational and learn to do bioinformatics sort of stuff. But the problem was that she didn't really have any experience of that. And so we talked, and I was like, in the end, I sort of issued her a kind of challenge, and I said, I think you're really good, and you can join my lab. But the thing is, if you want to learn to do this with bioinformatics, you're gonna have to teach yourself. You're gonna have to learn this stuff. We're not really gonna be able to hold your hand too much. We got things to do. We got problems to solve here. We don't have time to drag you along. So she kind of she took the challenge, and I'm just delighted to report to you that she rose to that challenge. She taught herself all kinds of stuff. She sent herself to Cold Spring Harbor and took a course there, and now she's really good at this stuff. And I think her thesis really kind of illustrates, to me, your thesis looks like what a modern thesis should look like. It's about half computational, it's about half experimental. She did the experiments, she did the analysis, she draws the hypotheses, she does the analyses. It's just a lovely example of what a student can do when they put their minds to it. Her New Jersey attitude really was required in this project. We have been working for a long time on models of gene expression. We keep telling ourselves that we're going to be building these models of gene expression, that we're going to be able to predict how genes go up and down based on DNA sequence. And Dana came and she was on board with that. But unlike all of the previous graduate students in the lab, Dana actually decided to put this idea to the test. She wasn't really satisfied with just model building. She actually, I think, was really the first person after about 10 years to actually decide to test some of these models. And that took guts, which she has in spades. And that was necessary because it didn't exactly work out the way we thought it was. There were aspects, as you'll see, there are aspects of these models that really work beautifully. And she's going to show you some of that. I think she's going to show you some of that. And then there were other parts where our models didn't really work kind of the way we thought they were going to. And this was a part where it really took some perseverance, some creativity, some imaginations to kind of take the lemons and make lemonade. But in the end, I think it's a nice story. I think it's a measured story. I think it's true. I think it's a great example. It's another great example of what you can do when you actually believe your data. The result was a little wonky, but we never thought for a second that this had anything to do with the quality of the data. Dana's work is well, she'll show you. I think she used to tell me she wanted to make a T-shirt that said something like, "What's your R squared?" We'll leave the last word out. I think it was a New Jersey word. Anyway, I'm excited to introduce her. She rose to the challenge. I know there's going to be more challenges ahead. If you meet them in any way like you met the challenges here, you're going to be absolutely fine. I'm excited to see where you go. Thank you for joining my lab. Dana King.
It's really funny that Barak talks about that story because that's like in my acknowledgments too. His exact quote was, "You'll have to do the work." And I was like, I'm not sure what that means, but okay. All right. One of my committee members is telecommuting in, so this is what that is. So I just need to set that up. No one will get the pointer. It's okay. All right. So thank you for that really kind introduction, Barak. It's been a pleasure to work in your lab, and I'll talk more about that, I guess, after the sometimes beautiful data. I'm happy to share my thesis work with you. It's titled Grammar Variation, Understanding How Cis-regulatory Information is Encoded in Mammalian Genomes. So getting to this title was a long journey. And if it seems a little complicated now, don't worry, I'm going to break it down. So the first question I think we need to answer is what kind of information can be encoded in a genome? So we know that genomes are made out of DNA. Parts of that DNA makes RNA. And then some RNAs go on to make proteins, so those carry out important functions. And those functions are critical to defining and allowing for proper function of a cell type. And we know that many, many different cell types make up a whole person. So the function of the cell and the phenotypes that we see as complex organisms is dependent on the DNA sequences that we have and the function of those sequences. So the next question is, how is information encoded in the genome? So I show it as this physical diagram, but we can also think about it as a string of letters. So these are the nucleotides along here at this very, very small scale. But the genome is really, really big, so we can also think about it as a string in some ways. So this physical area is composed of different functional regions. So one part that I already told you about is the coding region. So these are genes that can make proteins or non-coding RNAs. And then all the rest of the parts we can summarize as non-coding regions. So non-coding regions, some parts we can designate as cis-regulatory sequences. So these are the portions of DNA sequences that are critical to controlling the when and where a gene is turned on. And these come in two flavors. So one is the promoter, so that's thought of as being usually close by to a gene. And then enhancers are usually very far or very, very far away. So these are all, I'm going to abbreviate this as CRSs. And then within these functional cis-regulatory elements, we have these special sequences called transcription factor binding sites. And these are DNA sequences that help recruit proteins that carry out the functional work to turn on a given gene. And so with these sequences, the ability to recruit proteins produces the RNA that's necessary to go on to make a protein. And this process is critical for development in disease. So if the gene is not being turned on in the right place at the right time, you can't develop. If it's suddenly turning on too much, that's how we think about how cancer happens. And there's a whole host of examples that I could give you, but we have a lot of slides to get through. So the big question that my work has focused on is how do differences in sequences impact this regulatory activity? So we know that these sequences are in the genome. What happens if these are changed? And I have two parts of this story. The first part is grammar in mouse embryonic stem cells, so MEMCs. And the second is variation in this human lymphoblastoid cell line, which is an immune progenitor. And I'm showing you these little organism cartoons, but the work was actually done more like this, where it's cells from these organisms in dishes. So that's a little bit of a caveat. We're not describing the function of a whole person or a whole mouse even. All right. So for my first project, the proximal question I was trying to answer was why do we see different levels of expression from sequences that look pretty similar to each other? So I'm showing you transcription factor binding sites here, close to a promoter, different levels of RNA. And these have the same sort of color, so we would expect them to recruit some of the same proteins, but they're not quite the same. And the observation that we see is that they then drive different activity. So one way to formalize this is to define the arrangement and other attributes of these sequences in regards to the transcription factor binding sites as grammar. So here I will define grammar first as composition, so what colors are there, what proteins can be recruited to a given cis-regulatory element. Next part of the definition is position, so is it close to a promoter, further away, and that. Next, orientation, so is it are these binding sites facing forward or away from each other, maybe changing the different protein-protein contacts that can be facilitated. And similarly, what is the spacing between the sites? Are they so close together that they might impede the binding of one another? And then lastly, an attribute of transcription factor binding sites is affinity. So how close to the ideal binding site is this piece of sequence? And then for my project, I've really been asking, or for this project, I've been asking, does grammar matter for pluripotency factors? So you have to choose what transcription factors you want to look at, what binding sites you want to look at. And mouse embryonic stem cells are a great model because they have 4 plus or minus like 3 transcription factors that completely define the state of the cell. So with these transcription factors, you put them into another cell type, you can drive it towards this pluripotent state. So the hypothesis that's been driving this project is pluripotency factors do have some specific interactions that require favorable configuration, so requires some sort of grammar, agnostic to the exact flavor of the grammar. All right, so one way that we went about testing this is using synthetic elements. So these are sequences that we can control exactly what binding sites are present, and then all possible combinations of arrangements that we're interested in. So I made them of like the 4 binding sites, so one of each of the factors binding site in the sequence, and then 3 and 2, and then I have a set of controls where there's no regulatory sequence included. And so this allows us to test position and orientation while fixing spacing and affinity. And in the Cohen lab, we have this really powerful tool to test large collection of sequences very precisely. And that's called massively parallel reporter assay. So the way this works is we put in a sequence of interest, so synthetic or real genomic CRS, and then we match it up with a DNA barcode that's going to be downstream of a reporter gene. And this barcode allows us to count the number of RNAs that this sequence of interest then drives, and we can compare the number of RNA barcodes against the number of DNA barcodes, take an average over however many barcodes we decide to assign to a given sequence for redundancy, and then we can calculate the expression of the CRS. So I did this for those synthetic elements, and first what we see for the data here is number of binding sites, here is expression, and the first thing that we see is that adding a binding site boosts expression. So these sequences are pretty active, they're pretty far away from this basal, which has been no sequence control. Add a site, you get more expression. When you add an additional site, you get another boost up here, so these sequences are on average more active than these sequences. But we also see this really interesting pattern in these formers. They have the same exact identity of binding sites, but their composition is, but their expression is very different. So that has to be due to the position and or orientation. So next part of my analysis was to ask that exact question of, do both of these things matter, or is it just one part of the variables we're allowing to change? And so in the next slide, I'm going to summarize the binding sites for these factors in single letters. So capital K is forward, lowercase k is reverse, and that's the same from all the other letters. All right, so first I looked then at the low versus high synthetic former, so for that distribution. So here again is the mean expression that I calculate, and then plus a standard error, the error bars. And on the left side is the lowest sequences. So we can see that many sequences drive low expression. They have some differences in the pattern, but they all are pretty equivalently bad at driving expression. And then again, versus the high guys, where again we see many that are driving similar level of expression, but they also have some similar patterns of sites. So we see that SOKE is overrepresented in the highest, most expressing guys, and the orientation doesn't seem to matter much, where this SOKE is about the same as this SOKE. So this suggests that the position matters more than orientation. So how can we formalize this? Barak spoke briefly of the quantitative component of my thesis, and one part of addressing the individual contributions for the parts of grammar was to build models. So the model that we settled on for various reasons that I can talk about later is a random forest model. So this is a machine learning approach that allows us to take any feature that we want that describes a given sequence. So over here, this is position. So where is each of these binding sites? And I can also specify, is a binding site present at all? So that independent contribution of adding additional binding site. And I trained a model with these features for the composition and position of the binding sites. I'm going to show you that performance on the next slide. All right, so here is the observed expression of this portion of the library, and here is predicted. So I'm showing you the training data here. We get a good agreement between what we observe and then what the model predicts. And this did cross-validate. So that's great. So there's something about the features that I'm putting into the model is describing our data. So what does the model say as far as the contribution of those features? So because of the type of model I'm using, it weights the different variables, different features that I'm putting in. So we see that adding a K, the presence or absence of a KLF site, is really, really important to the model. And then it's in agreement with what we see for the top and bottom most formers. We see that having a SOX in that first position and that ESRB in the last position is even more important than adding an after SOX or ESRB independent position. So that was really exciting because we can fit our data and we can formalize the type of grammar. So for synthetic elements that combine these potency binding sites, what did we learn? So we learned that composition does matter, so different numbers of binding sites have different expression. We show that position also contributes to the quantitative levels of expression for these sequences. Orientation doesn't seem to be clearly contributing, but I would be cautious about completely ruling that out, and then spacing and affinity are not applicable here. All right, so this is great. We fit a model, but these are sequences that you're not going to find in the genome. So they're highest affinity possible. They're just like the ideal binding site. They're fixed distance from each other. That's not what we see in the genome. So the next question I tried to ask is, does the synthetic grammar model apply to the genome? So when we have now variable composition and variable orientation and position and spacing and affinity, when everything's changing, do these two features that we see contributing to synthetic library help us explain what we observe for genomic sequences? So first I had to find some genomic sequences to compare to. So I found 400. So I looked under peaks for the factors of interest, identified binding sites that were no more than 20 base pairs away, and then picked sequences that had 3 sites per element with no more than 1 site within that distance. So as similar as possible that I could find for the synthetic elements. And then I tested these, again, using MPRAs and mouse embryonic stem cells. So these are comparable data. So I'm going to show you the performance of the synthetic model on the expression data from this collection of sequences. So the axis is flipped, so sorry. On the x-axis here is predicted expression, so this is prediction from the model, and here's the observed for the genomic sequences. So these are all the features that went into predicting, so retraining the model. This red line is what the x equals y axis would be, because these limits are very, very different. So even if we kind of scale this out, stretch it out to be the same axes, the model is not very predictive. I forgot to look at my R-squared, but it's something around 0. I think it's like 0.001 or 0.007. So that tells us that composition and position probably, maybe don't contribute strongly, or at least not in the way that for the assumptions that we have for the synthetic library. So one reason that the composition doesn't contribute is maybe the activity of these sequences don't depend on the sites at all. So I also tested a set of mutated sequences where all the sites that I identified as pluripotency binding sites, factor binding sites, I mutated. So it's a 1-to-1 match. And I tested these using MPRAs. So what does this data look like? So on the x-axis is the mutant expression. On the y is that wild type, so what I showed you before. This is the control, so things that are not active in either mutant or wild type will be right around here. And then our expectation would be that if the composition doesn't matter at all, the mutant and the wild type expression would be along here because it would be independent of the binding sites. So thankfully, we don't see that at all. So we see a lot of inactive sequences regardless of if they have the binding site or not. So these are in gray. But for the sequences that depend on the site, so significantly different between the two, we see that they're way off the line. So this suggests that the genomic sequences do use at least one or more of the sites. We can't distinguish between the two. So if there is a grammar, it should depend on the site. All right, so next I asked, well, if it depends on these sites, then what sort of grammar might apply in the genome? So composition, there could be other binding sites, so these matter, but maybe there's something else contributing. Position orientation, not sure. What about spacing and affinity? So I don't have time to go through all of the things that I did in order to try and get at the grammar for these genomic sequences, but I will tell you about the binding site affinity and how that contributes. So to make this easier, in that we don't have a lot of sequences that are active, and they're really, really different, I made this into a classification problem. So instead of trying to quantitatively model this dot versus this dot, I grouped them together into low and high. So what describes these sequences together? And I asked for each of the binding sites, does the affinity, so how close they are to this ideal sequence, matter? So I'm going to show you that now. So this is the affinity score, so how close it is to that ideal site. And the numbers here are not super, super important, but here's the low versus the high bin of sequences, the average affinity score. So for SOX2, it doesn't seem to distinguish between high and low sequences, which is interesting because SOX is a really important regulator in ESLs. But it does seem to matter for the other three factors. So OCT4, which is a binding partner of SOX2, the affinity of the site is significantly different, as it is for ESRB and KLF4. So these distributions are fairly wide, but it does appear that this could contribute to the differences that we see. And again, I don't have time to tell you the details of this, but I did find that there are other factors that likely contribute to driving low versus high expression. Position orientation through modeling does not seem to be supported as part of the grammar. And we do see some signal for spacing and for this affinity that I just showed you. All right, so what type of grammars do they have? So here we learned that the synthetic elements, when we control spacing and affinity, we see these effects for composition position. So, but when we look at the genomic sequences, composition does seem to matter, but we don't see the effect of position or orientation, likely because this is a small effect that is washed out by the site affinity. All right, so this is the first half of what I'm going to talk about. So the next part of my thesis has been all about testing variation in this immune-ish progenitor cell line. And an important piece of information for this project is the fact that different people have different genome sequences. So if we look between a population, between individuals, there's variation in both the coding and non-coding regions of the genome. So there's variation. We know that. There's been huge efforts to catalog this. And we know that that variation can have consequences. So I'm showing you here a gene-wide association study, so looking at individuals, either classifying them as, in this case, immune disorder or not immune disorder, and associating the variation that's different between those groups in a very oversimplified summary. And we can see that the coding region, which is all the proteins that do the work of the cell, that's only 10% of the associations. Enhancers alone is 60%, with an additional small amount for promoters. But everything outside of that coding region we can classify as non-coding. So it's likely that non-coding regions contribute to disease in some fashion. this is great to know that, but it creates a really hard problem. So for figuring out what the function of non-encoding variants is, we have a huge genome. So if this was printed out on a phone book, it would be 200 phone books and 1 million pages. So that's a lot for each person. So times however many people you need to get an association. All right, and then the catalog of published GWAS studies has about 75,000 associations between individual variation and some trait. So, and those variants are often very far away from coding genes, so it's hard to assign what they're doing and in what way. So one solution that has been an important part of the field is using predictions to try and prioritize variants that might contribute to cis-regulation. So what can we use modeling in order to address this problem? So there's been two main ways that people have approached this previously. So using functional annotations, such as from projects like ENCODE. So can we bin the genome into its different functions? Or using population genetics for conservation. So population genetics being the differences between individuals at different locuses, or conservation, so how little has something changed over evolutionary time by comparing species. Recently, though, there has been efforts to combine these two types of data, or these two types of predictions, into a single measure. And the two approaches I'm going to tell you about today are called FitCons and CAD. So FitCons is developed by Adam Stiefel at Cold Spring Harbor, and it claims to predict mutation-sensitive cis-regulatory elements. So which ones won't be happy if there's a change made. And they describe it as a probability of mutational fitness consequence. And the other algorithm is called CAD by Jason Jury's lab at University of Washington, and it claims to prioritize functional cis-regulatory variation. So they describe it as a measure of deleteriousness, so deleteriousness is bad, which strongly correlates with molecular function and pathogenicity. And this is got messed up for some reason, but this should show the genome with the variant. So can we get functional annotation, combine these two pieces of signal, so evolutionary signal and functional annotation, and enrich for cis-regulatory sequence? And the hypothesis that has been driving this part of my thesis has been predictions that include regulatory and evolutionary signals will help predict the impact of variants. So we came into this thinking that these algorithms would help. Okay, so what did I do? First, I found some sequences that were putative regulatory elements, and I looked into the 1,000 genome database, which is just a collection of healthy people, kind of as a goal to use as the baseline for our expectations. And for each cis-regulatory sequence, I tested 350 reference and about 350 alternate alleles, so a single substitution for a given cis-regulatory sequence. And the distribution of scores for these sequences for FitCons is about 165 low, so low probability of consequence to fitness, and then 175 that were at the highest end of what we see for the non-coding genome for these predictions. And then for CAD, it wasn't quite an even split, but have about 200 of these same exact sequences. The annotations are that 200 are about low, and about 140 then are high with the CAD score. All right, so I took these sequences, which have two different prediction sets, tested them in these lymphoblasts cells, and then did the same sequencing and calculation of expression for each individual reference or alternate CRS. All right, so what does this look like? On the x-axis here, I have my alternate allele, so this is when you make a change. And then on the y-axis is the expression of the reference allele, so what's in the reference genome. And this red line is the XY axis. This is what we expect if the substitution doesn't matter. And we see that these dots go all the way up these lines. So there's a wide range of activity for the sequences that I tested. And we also see that most of them really cluster around the line, suggesting that there's not a lot of big changes. We do see stuff coming off. And we are able to find variants that are significant, and those are significantly different between alleles. So that's these blue dots. And to summarize all this data into a single measure to compare with these predictions, I calculated an allelic skew. So that's the expression of the alternate allele, expression of the reference allele, and take the log ratio of that. And because we're not really interested in what direction it goes, I took the absolute value of this, and that's what I'm going to show you in the next few slides. All right, so the first thing that I'm showing you here is, despite our hypothesis, allelic skew, which is on the x-axis here, the absolute value, doesn't seem to be different between groups of low fixed-cons predictions and high fixed-cons predictions. And even if I just take away the quantitative level and ask how many of the sequences in each of these bins are significantly different at all, that's also not significant. So if there is a signal, it's not very strong, given the size, the number of things I'm testing. All right, and then what about for CAD? Maybe CAD is a better algorithm. They were generated independently by different groups from different data sources. And so allelic skew is again on the y, and lone CAD and high CAD also don't appear to be different, statistically at least. So, and the same is true that even if we take away the quantitative level, there's no difference in the number of significant sequences with allelic skew. All right, so this work is still a little bit in progress, but I think we've learned some important things. So as far as what variants will impact this regulatory activity, prediction by these combined approaches, for 1,000 genome variants at least, many of the variants we test will drive allelic differences. They're small effects, but FitCons or CAD does not predict which sequences will have an allelic skew. So if there's small effects, do those small effects matter? I think that's an open question. All right, so going back to my big question, how do differences in sequences impact this regulatory activity? So I hope I convinced you that at least in mouse embryonic stem cells, when we manipulate the sites, the transcription factor binding sites and their arrangement and sequences, we can see that synthetic biology works. So we can fit a beautiful model, but it doesn't seem like we can apply it at least directly to the genome, which makes sense. They're very different. And I think learning about the grammar can help us understand the possible interactions between the pluripotency factors to understand how they might function in the genome, especially where a lot of these were active, but in my genomic sequence, a lot of them are inactive. So is there something about how they interact with each other that we're missing because of the way that we think about the proteins? All right, so two main, I think, future directions for this part of the project would be that design synthetic libraries that are more representative of the genome. So I showed, at least for pluripotency factors, that changing affinity is really important. So designing synthetic libraries that change affinity might give us a better handle of what to expect from genomic sequences. Or changing the spacing slightly, so not fixing that. And just tuning those two things outside of maybe just ignoring orientation. We don't need the sites in both directions, at least for pluripotency factors. And then the genomic library wasn't as active as we expected. So is there a role for maybe repression? So I think it would be really cool to modify MPRAs so that they pick up on this little teeny tiny bit here that's below basal, below having no regulatory element, to see if that is true repression. And then for the second part of my thesis, I think we learned that predictions that incorporate functional data and these evolutionary signals, they could be useful, but at least for the alleles that I tested, in the cell type I tested them, they don't enrich for allelic differences. So they might have value, but it's hard to know how we should approach figuring out what those predictions mean versus what we expect them to do. And I think how to predict variants that do impact this regulatory function is an open question. So it would be really, it's going to be interesting, I think, to see in the next few years like how different approaches measure up to each other, or if MPRAs are the right way of going about testing this. And just for the immediate future direction, so until I move on to somewhere else, what I really want to know is, at least with what I've tested here, what features of the sequences do help us predict expression differences. So I have a rich set of data, and I have only really skimmed the surface of it. And then are there other ways, other computational tools that might perform better for these data? Okay, so I'm going to do some acknowledgements, but I don't think I have a ton of slides. So first, I have to thank my thesis committee. Joe recently joined. Jim was in a lot of the meetings, but he's not here right here. And of course, and Kristen is remotely. So thank you all for your inputs. My work would not be at this level without you. And then for sequencing, which is the entire basis of all of the data that I showed you, Jess in the CGS has been invaluable. I just show her my things. She tells me what other people in the lab had done, which Barak won't know, and then she makes me have beautiful data, and then I can analyze it. And similarly, Brian and Eric, if I run into computing time issues or coming from absolutely no experience of using cluster, ask what does submitting a job mean. They explain it to me without making me feel really dumb, so that's great. And then my projects have been funded through the NIH. I meant to add which R01 it was, but Barak was out of town a lot this month, so. And speaking of Barak, I'll start off with him for acknowledgements. We like to think of him as the captain of our intellectual ship, and he has really been a great mentor. He will come out into the lab every once in a while and touch experimental things, but he recognizes that that's not his primary role. He's not out here trying to tell us, well, like for this PCR, this is what you should do. He's like, well, let's think about it. Let's think about the problem and how to address it. And he has a great sense of humor. So this is a prank that we pulled on Barak, where we wrapped everything in his office with Trader Joe's and Whole Foods bags. And he was pretty happy about it. Like, took him a minute. He could still sit and do work. He said he hated Trader Joe's. So we brought Trader Joe's to him. And I guess for Barak, it's so funny that he introduced my rotation that way, because I came into my rotation really excited about the type of work I was doing, which is completely unrelated to any of this, but really intimidated by the amazing people and by Barak too. I was kind of scared of him, which I feel like is what people think of me, where they're like, you're really scary. And I'm like, I'm so nice. He's like, why are people scared of me? Maybe it's the like New Jersey, New York kind of connection. But I remember him distinctly saying where I'm like, I really want to join this lab. I'm really excited about it. I tried to write an ordinal differential equation model, which is hard, in 2 weeks of my rotation with no prior coding experience, and I failed. And I thought, you know, I'm never going to be able to do this. And Barak just is like, he never told me my rotation was good. He was just like, yeah, I mean, you could join, but you'll have to do the work. And I'm like, well, I'm going to do that. I'm going to show you that I can do this. So it's pretty amazing how far we've come in communicating with each other, and I've just grown so much as a scientist and a person in knowing Barak. And I also need to acknowledge our great lab. So this is the current iteration. I've been here almost the longest out of everyone right now, outside of Mike and Barak. So it's gone through a few iterations, but this is from our most recent picture out of the side of our beautiful, beautiful new building. And I think it just shows kind of like both like the warmth and the congeniality of everyone in the lab, that we support each other, we challenge each other, but we can have some laughs at the end of the day. And this group and scientifically is great, and as a support system has all been great. So I wanted to include some like previous members of the lab that I overlapped with. Mongi was my former Baymate, and she graciously let me bother her with all my art questions. We won the best tasting dessert last year through great teamwork, representative of our scientific teamwork, because they're basically the same. We can have a good time, and within our community, we're also part of the CGS, which has also been just the best environment I think I could really hope for. So this is from a hackathon and some celebrations, but just trying to highlight. I didn't have many pictures of CGS, which is kind of weird because I spent so much time there. But just the community that we have, I think, means that we can do great science and still be happy, which is not, you can't find that everywhere. And then I want to also acknowledge my grad school friends. So Sarah and Caitlin, I think, are here. Kelly has moved on. Rebecca is letting me live with her because I'm between apartments. So really, can you do any better than that for friendship? Shalina and Julie, I think, might have popped in here, but I'm not sure. But just the people that I've met through grad school have been invaluable for moving forward and being able to do what I have to do to accomplish these things. And then outside of grad school, I have a lot of people that have helped me get here. My roommate would walk my dog when I would be getting home from lab too late because experiments aren't working. So he would walk my dog and laugh at me when I told him things about my project. And it's just like, you're so impressive. I'm like, that's not my day-to-day experience. So thank you for appreciating me that way. And then I have some great friends that I know are watching remotely, who I met when I was a baby scientist teching at Coulter Harbor, and they have just supported me on every part of my journey, moving out here. They actually came out to St. Louis for my birthday, and just have been the most wonderful and like really sharp as tacks women that I could have as friends. And along that lines, I also I don't have a lot of good pictures, but this is kind of the best that I have of. We have a reading club called Rage Pages that April is one of the people driving the group, and it helps us talk things out, get some career strategy advice, vent about how it kind of sucks to be a woman in a male-dominated field sometimes, and that you're not alone, and thinking about kind of the world and the context greater. And then I have some other great friends that I met, Julia and her wife Bonnie, that have really showed me how to show up for things. And then Barak showed this picture of me and my friend Deanna. She has also let me stay with her every Christmas, so I can take a break and relax in New York and not have to explain things to my family. So, and she came and visited also and was there when this is my first showing that my first EF experiment was going to work. It hadn't worked, and then it was. So she got to witness that. And then I didn't have a picture for my friend Kat, who also let me live with her, but her little dog, Indy, and my dog, Theo, have been best friends since they were at daycare together. And she does biostats, so we can talk about work, we can talk about those sort of things, and we can also just relax and watch our dog play and take a break from all of that. I also need to, of course, acknowledge my family and my boyfriend Kevin's family. So this is from, I think, Thanksgiving last year, with our dog in the center, of course, and me in the center, which I didn't realize I did that in pictures until Mark showed me. So they've just been so welcoming and so supportive. Vicky's here today, so I told her about 2 weeks ago, I think, I was like, it would be really nice if you could come to my thesis, and she's like, I'll be there, no problem. Just dropping everything, coming here, and just really being curious and just really, really warm, wonderful people that I have a privilege to know. And then I have my sisters. So my sister actually had a bone marrow transplant when I was a kid, and she is super healthy, so don't worry, it's a happy ending. But because of going through that experience, it really got me interested in genetics as a field and learning about Punnett squares, and I'm a healthy non-carrier. She's a healthy carrier, but unaffected. So I like to joke around about that with them. They don't always understand, but they have supported me, like, forever since we could be cognizant, and we've gone through a lot together, so it's really great. I think one of them might be watching, but I'm not 100% sure. And then my extended family, who really encouraged me when I do go home and push me to do more. And then last but not least, because he would be really upset if I said least. Kevin, my boyfriend, has just been, we've gone through a lot through the process of grad school. We met here, and he has moved out of state, but he was really excited at the beginning, probably a few months in us dating, and he thought I got a good deal. Then he was scared because he met my friends, I think, and he was living in Seattle at the time too, so he'd been taking red-eye flights. But this is us just a few years ago. I think we've mellowed out to a good place, and he's a partner who I can really talk to about my work, about what I'm going through, and is just a supportive and incredible person that I'm really privileged to know. All right, so I'm only 3 minutes under my expected, so I didn't talk too fast, but I would love to take questions now before we go to the closed section. And thank you all for being here. Yeah?
So your synthetic library doesn't really have spacing for the test position. Should sort of use the spacing in some way?
Yeah.
Have you tried to use anything from that to map back onto your genomic library?
So the thing with the genomic library is because it's a continuous scale of spacing, I could see and I saw, I'm not going to pull up the slide unless you really want me to, but it's basically you see the helical turn. So with the synthetic, they're fixed on the helix. So if the orientation was a strong effect for the synthetic and it was a strong effect of spacing in the genomic, I think that would work. I did try a bunch of things to try and make the genomic more synthetic-like, or the synthetic more, well, you really have to go that way because the data is the data for the synthetic. But it's really this pretty diffuse, small effect. So the genomic sequences, I guess a better summary for that would be, we see a lot of things with small effects, and I have 400 sequences. So I think if we tested more, maybe we would have more power to kind of map it back or forward. But I did think of that and tried something along those lines, yeah. Other questions? Yeah.
Lovely talk. So at the start you defined affinity in terms of the distance from the complete binding sites.
Yes.
Do you have a sense of how that definition of affinity relates to sort of real KD biophysical affinity? Because is it possible that SOX2 is just more promiscuous, for example, even though those two things are different, maybe they're binding the same strength?
Well, SOX and OCT are known partners, so yeah. So I guess your question is, how good are these perfect ideal sites compared to what we would expect for the biophysical? There's not good biophysical models of SOX and OCT binding. There's just the DNA binding domain, so not the whole protein, and we know they have protein-protein contacts. So first pass. Also, the binding site that I used to score everything, they're a little bit older, so there's better versions. Actually, we have a collaboration with a group that developed really much more quantitative binding site predictions. And I chose these sites because they were what we use for our synthetic library, so at least for comparison. But I would say they probably don't represent the biophysical very, very well, but it gives us a sense of, at least as far as sequence composition and however the proteins are reading that out. And I think for SOX, it might be that SOX can bind or not, but if it doesn't have OCT, maybe it can't do what it needs. And I do think that there's probably some things that we're missing as far as when it's now being repressive, because OCT can also interact with repressor chromatin modifiers, which is a whole thing outside of this, another layer of complexity. Yeah. Other questions? I guess I talked too fast. understandable. Thank you.
Okay, so regardless of the outcome of the next hour, we will be gathering again at about 1 o'clock on the 4th floor in the Couch Building. You're all invited. Thank you.